Excellent. There you are. Very well. All right. So let's start. Let's get started today. We will talk about the nucleic acids. Nucleic acids. Okay. When you're talking about nucleic acids, don't be afraid about the word nucleic acids, right? These nucleic acids means nucleic means nucleus, right? Acids because those are actually we have the RNA and the DNA that actually have that behavior. So that are the element, the DNA and the RNA. In conclusion, what is a nucleic acid? Acid is the components that are in the nucleus, nucleic. So those elements are called the DNA and the RNA. So what are the nucleic acids? RNA and DNA. We okay with that? Hello? Yes?
Yes. Yes.
Okay. All right. So I'm going to go into chromosomes and genes. So it's kind of probably in the past was very confusing or slightly confused. And my purpose today is to basically try to clarify all these elements. Okay. So chromosome versus genes versus DNA. All right. So let's start. So I will say this. So look at this. We have a cell here. This is a cell. And we have the nucleus here. Okay? We have another cell who are going to contain, this is, I'm going to put here the DNA. The DNA. I'm going to put the DNA here, drawing here. DNA. This is DNA. And then we have these ones here, like this. What are these called? What are these little things called?
Cytochromes.
Chromosomes.
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 And that is what we call the DNA, correct?
Yes.
Okay, so tell me, if this is a cell and you have DNA and chromosomes, tell me, this portion that you have in the nucleus is the same as this, what we have here, chromosomes? Yes or no?
Mm-hmm.
No.
No.
No, no, yes.
No.
No.
Yes.
The answer is no and yes.
Oh.
It's no and yes. All right. So why is that? So the DNA here in this state, so there is two different stages. So this stage, basically this is exactly the same as this. But in appearance is different, of course, right? But the content, what we have inside the DNA are exactly the same between the DNA and the chromosomes. So when is that happening? Very simple. Number one, here we have in the stage one, here in this period, the patient, the cell are resting. And here in this stage, these cells are multiplying, are dividing. So this all together, the appearance is different, but all the content, all what you have inside is exactly the same as the double spiral DNA of the DNA. You okay with that? Okay.
Yes.
So what happened here? What happened is that this DNA, what it's going to do is to, when they are going to divide, you have one DNA here. So in order to have a cell division, they're going to divide into cells, correct? So this DNA, they need to multiply itself. So you have two DNAs here. Two DNAs. Because the cell is going to multiply. And the multiply means that you have two cells that are going to have equal number of DNA. All right? So now, this, basically what it's doing is this. They're going to uncoil. And these uncoiled DNA are going to be like a thread. Then these threads are going to coil by itself. Correct. So they're going to actual coil. And they are going to cut in pieces, cut in pieces, cut in pieces, 46 pieces, 46 chromosomes. And that's why they are going to, each piece is one of these. This piece is one of these. This piece is one of these. This piece is one of these. So all pieces together, if you make a summation of them, they are going to give you the double helix DNA. You okay with that?
Yeah.
Yeah.
Okay? All right, perfect. Is that clear, please?
Yes.
Okay.
Yes.
Okay, so now here we have a chromosome. A chromosome is a part, a piece of the DNA. I want to tell you something. You have 100 trillion cells in your body. I repeat that many times in the past. But let's make it for some magic procedure, from some magic instrumentation, whatever you want to call. You can put your hand on one of the cells and take the nucleus and take the DNA. The DNA, if you make like stretch out the DNA, if you can stretch out the DNA, stretch out the DNA from each cell, that is going to give you 6 feet long. Can you imagine that? 6 feet long. And you have 100 trillion cells who contain this strand of DNA. Yes, literally. If you the length of the DNA is going to be about 6 feet long. Okay, if you uncoil it and stretch it out the DNA. Okay, so then we have another thing is going to be, for example, the genes. What are the genes? And the genes are the segment of the DNA. I'm not going to be a more introduction about this, but I want just to see this. All right. Hello. Okay, Mr. All right. All right. You okay? All right. All right. I want just to think about this. Look at this. Follow, please, my slides. So I want you to pay attention on this because I'm going to make a summary to make it easy for us to understand. Look at that. Here, this is the DNA. The DNA looks like what? The DNA looks like spiral stairs. Yes or no?
Yes.
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 Excellent. Okay. So all this is like a spiral stairs. So I'm going to translate into this. Okay. So here we have the DNA. The DNA, imagine yourself, you are actually standing up here, standing up, well, okay, whatever. Okay, standing up, and you are going to walk over the stairs. These are steps, correct? These bars are going to be these lines are going to be the bars, and this line is going to be the rails. The rails. That is exactly the same what happened with the DNA. The DNA helix are going to have these steps. They are going to have these bars, and they are going to have these rails. Okay. Let me go here just a moment. Okay. And here we have those steps are going to be called for DNA. Now you're going to call base, a base. That is a base. So imagine a step on the stairs, and where you put your foot on these steps, that is going to be called a base. Base is what we have all the sequence of many elements that you are going to learn in a few moments. Okay. The bars, these bars that you see here, are going to be like sugar. Sugar is going to be the deoxyribose. And the rail are going to be the phosphates. 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 All right. So please, I want just to imagine yourself going into a stairs and close your eyes and put your foot on the first step. That is called a base. You are walking on the DNA right now. So you are closing eyes and you need to support. You're going to hold the rail in order to help you to climb, to go up into the stairs. What you are touching there is the phosphate. The rails are the phosphate, and the connection of the rails with the bases of the steps are going to be called the sugar. Sugar. So we have base, sugar, and the phosphates. You okay with that?
Yes.
Okay.
Yes.
So that is about the um, that is about the uh, configuration of the DNA. The DNA, if you have a human-sized body, the DNA, the bases have a human proportion size. They're going to have a building that is going to measure about 1,000 story. 1,000. There is no in the world that has 1,000 story building, and that is the DNA. So do you have about 140,000 steps? So that means that you have these bases 140,000 times. All that. You okay with that? Okay.
Yeah.
Okay. Now, what I want just to go on this, let me see if I have another picture here. There you are. All right. So the first picture. So here we have the stairs. Imagine you see here this yellow and red you see here, that is the bases. The bases. This ribbon that you see like a white undulating here, those are basically the phosphates. And the bars are attaching the phosphates with the bases. Okay? Now, what is a gene? What is a gene? That is the moment that we need to know what is a gene. A gene will be this piece. For example, forget about this for a moment. We have the DNA here, the DNA, and the DNA, they have like 1,000 story building stories, right? Levels. So you will see that a gene is a piece from here to here. What does it mean? Imagine you're going to the stairs and you're climbing up the stairs, and you go from floor number 3 to floor number 20. So all these bases, all these sugars, and all these phosphates that are in that interval, in that from the third to the 40 story, that is called a gene. That is called a gene. So another gene could be, for example, go from a story number 10 to 12, very small gene, or it could be from story number 80 to 120, and that area, all this is called a gene. So the DNA is going to be composed by pieces that are called the genes. What are inside the genes? Inside the genes, we have the bases, the steps, we have the rails, the phosphates, and we have the sugars that are the bars. You okay with that? Is that clear or not, please?
Yep.
Okay, so don't forget that. So that is a gene. Now, what is a chromosome? The DNA that is a lone line, they are going to be divided in pieces, and these pieces are going to contain genes. So each of these chromosomes at the end is going to have a certain number of genes. Again, so let's make it simple here. We have the DNA here, the DNA, that is the DNA. Then the DNA is going to actually uncoil, and they're going to be like this. Okay, well, I'm going to make it simple. We have the DNA, and the DNA is going to basically cut in pieces. Boom, 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 pieces. Each piece is going to be forming a chromosome. Each piece is going to form a chromosome. And but what we have inside these pieces? Inside these pieces, we have a certain number of steps of the stair, a certain number of bases, a certain number of phosphates and sugars. That is what is going to happen during the resting stage of the cell, and at the end, the cell division of the cell. Is that clear or not, please?
Yes.
It's clear.
That's clear. Excellent.
Yeah.
Okay. Yes, question?
Nope.
We Okay, we have about 24,000 genes. 24,000 genes. 24,000. So if you can imagine, close your eyes and imagine, go to the building 1,000 stories, and each section that is called a gene corresponds to, for example, floor number 1 to 10, from 11 to 25, from 20, 12 to 50, to 50 to 110. So all these segments that I mentioned, those sections are going to be called a gene. So the DNA is composed by by about 24,000 genes, so group of bases. Okay, we okay with that? Can I go to the next point? Yes.
Okay.
Okay. So now here we are going to see, if you see in this image, you will see here that this is the rails. Those are the phosphates that are around, and here inside the steps is where you put your foot to step on. That are going to be the bases. So these bases is actually what is going to give us all the elements that are going to build up in the body. All right, so I will tell you, just to make it simple again. So those bases, those bases actually are the recipe to for a protein. Listen to this very clear, please. Each gene, every single gene, is going to produce a protein. I didn't say fat. I didn't say carbohydrates. So each piece, each gene are going to have inside a recipe, written, written a recipe for how to make a protein. How to make a protein. So proteins in the previous class, you know that basically is everywhere. Proteins are in the enzymes, are in the hormones, they're in the receptors, are the immunoglobulins, are the hemoglobin, the myoglobin. So see, all these are being already established by the genes. So the genes will produce all of these proteins that we mentioned in the previous class. We okay with that?
Yes.
Yeah.
Okay. So now, saying that, I'm going to start going to the next level here. Okay. So let's make a DNA. Let's make one line of the. So I go into the spiral. I go into avoid the spiral. I'm going to make it straight. Okay. So let's suppose that this is the DNA, and the DNA are going to see. You're going to see the steps. This is one step, another step, another step. But the steps are forming half and a half. So that means that one half of the step is one side and the other half of the step is on the other side. So they are going to form steps like this. Steps. So imagine that the steps of the stairs are half is coming from one side and the other half coming from the other side. So but the step is still called a base. You okay? You okay?
Yeah.
All right. So imagine you are walking on those steps. Imagine you're walking on those steps. Imagine you're walking on those steps. What are these steps? These steps are made of some what we call the bases. What are the bases? The bases are going to be the adenine, the thymine, the what is the other one is the guanine, and the cytosine. Okay? So can you memorize that right now? Adenine, thymine, guanine, cytosine. That is what we are going to use now. Okay? All right. So adenine is going to be, for example, here. This is the base adenine, for one side, and the other half of the steps are going to be the timing. If you have in one side of the step are going to have the guanine, the other one is going to be the cytosine. Again, if you have cytosine in one side, you have guanine in the other side. If you have thymine in one side, you're going to have adenine in the other side. So always adenine and thymine are going to come together. Okay, and the other one is the guanine and the cytosine. So if you remember the names, can you repeat me with me? Adenine, please. Adenine,
Adenine.
Adenine.
thymine,
Thymine.
Thymine,
guanine,
guanine,
Guanine.
and cytosine.
cytosine. Excellent. So, for example, if I ask you what is the complement of the adenine, you will say the
Thymine.
guanine. If you tell me the guanine, it's going to be the cytosine. Or if I tell you the cytosine, will be the one. Correct? How to remember that? How to remember that is very simple. We have A, T, and T. The AT and T. So AT. If you have A, you're going to put T. If you have T, you put A. If you have A, you put T. If you put T, you put A. You okay with that?
Yeah.
And the cytosine and the guanine, another simple way to remember is this. Cytosine. And this is going to be another C, but with the G. See, very similar. Cytosine and guanine. So that's how you can remember these are complements to each other. You okay with that?
Yes.
Okay. All right. So now, this is important. So, what is the relationship between these bases with the formation of proteins? You need to remember that the proteins are going to be quaternary, before tertiary, before secondary, and before is the primary. And that is what we are going to talk today. So I know that there is kind of a gap here because what is doing the amino acids with this ATCG, okay? And that is like a secret code, a secret code. The secret code is telling you, for example, we have a sequence of three of these. For example, we have, I'm going to put here A, G, and C. That is a code for a type of amino acid. So what does it mean? That this is the amino acid A1, and this amino acid is going to link to this. Only one amino acid 1. So the code is coming by series of 3 of these bases. If I have AGC, that is going to be attached to this line is going to be called an amino acid. Another 3 are going to attach another amino acid. Another 3 is going to attach another amino acid. Another 3 are going to be attached to another amino acid. So at the end, you have a sequence of amino acids. But who is giving the order? The order is the DNA. And the DNA, how they give the order? By the sequence of the bases, the A, G, C, T, A, or whatever you are. So for example, you can have A, T, and T, AT and T. That represent that is going to call a type of amino acid. The next sequence is going to be C, C, G. They are going to come another type of amino acid, and they are going to link to each other and make a line of amino acid that is going to be called the primary protein. Is that clear or no, please?
Yes.
Yes.
Okay, so that is a relation between the DNA and the recipe to produce a protein. So the protein is a sequence of amino acids, and who is going to give you the sequence is the DNA. And in the DNA, who is giving the sequence? The bases. What are the bases? The adenine, the thymine, the guanine, and the cytosine. Be okay with that?
Yep.
Excellent. Okay. So now we are going to talk about something very interesting. Oh my God, it's almost 12. Oh, okay. So can we have a break? My God, I'm going to take some sugar. Oh, let's continue. Let's continue. All right. So we are warming up. This is the moment. Let's continue, please. This is the moment that we are going to talk about a very important how this is happening. All right. So please, here we have, and please pay attention to this. Yeah, with this we have our small break. We have here. I'm going to make a cell here. That is a cell. Okay. I'm going to make a nucleus here. I'm going to draw a nucleus. A nucleus. This is the nucleus. The nucleus is going to be, a cell membrane. So it's like you have a balloon inside your house. Your house is the cell, you have a balloon. Touch the balloon. That is a membrane. Inside of that balloon, we have the DNA. So the cell membrane, that we have the nuclear membrane, that is a double layer membrane. So what happened with this is this, that you have a DNA that is coiled like this. It's coiled like this. Okay, perfect. Here we are going to have some continuation, and probably you remember from your biology, and we are going to talk about another time, is called the endoplasmatic reticulum. The endoplasmatic reticulum. That is the endoplasmatic reticulum. We are going to mention here is the endoplasmatic reticulum. Okay, so what happened here? Here, what is going to probably make it bigger, the cell? Oh, okay, unfortunately, yeah. Okay, it's not a nice. Okay, so this is the nucleus, this is the endoplasmatic reticulum, and what is doing is this. Look at this. So here we have the DNA, and here is going to be the recipe for the genes. So we are not talking about cell division. Huh? In cell division, they are going to cut in pieces, cut, and that is not going to happen. So that is cell division. We are talking about how the cell are going to produce a protein. So, and the protein recipe is in the DNA. So what happened with this DNA? So we don't need all protein. We don't need the 24,000 proteins at the same time. They're going to just uncoil one piece of the DNA, where is the gene that you want to, or that you want to copy the recipe. Okay, so that is the DNA. And then what happened? We have what we call the RNA, in the nucleolus, the RNA. The RNA, what is doing is only one strand, only one line of bases, like this, only one line. This is coming here, and what it's doing is to copy all the sequence, yes, literally, it's going to copy all the sequence of the amino acids, all the sequence of amino acids, okay? So when they are being copied, all the sequence of amino acids, this RNA, what you call RNA messenger, the RNA messenger, okay, thank you, the RNA messenger are going to come out from the nucleus and they go to the cytoplasm, to here. So the RNA messenger that is in the cytoplasm, the RNA messenger, called RNA messenger, messenger, the RNA messenger, are going to be a copy of the DNA that was on the nucleus. Then we have the sequence of bases, for example, ATC, for example, CCG, another group of three, TTG, another group of three, AGC, for example. And all these are going to bring and call an amino acid here. Amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. They are going to be linked by the region that we already talked, and that is what we call the primary protein. The primary protein. The primary protein. Okay? So that is the what happened with the RNA messenger. I'm going to repeat it again to make a summary and review in order to make it clear. All right. So the process that RNA make a copy, make a copy of one of the genes of the genes of the DNA, that is called the transcription. Transcription. That is called the transcription. A process where the RNA are going to copy the DNA, that is called the transcription, TC. TC. And when the RNA messenger is coming out from the cytoplasm towards the cytoplasm, and they are going to make the sequence of amino acids, that is called the translation. Translation. That's it. Translation. What is translation? Translation is the actually the make the sequence are going to put together the sequence, a
Of amino
sequence
acid.
of amino acids. Okay? All right. So just to this is the first review. We are going to do more reviews in it because I want to be sure that you got the information. All right, so tell me. So there is two areas where it's going to happen all this process to produce a protein. The first portion or the first part is going to happen inside the nucleus. Yes or no? Yes.
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 Right? All right, so let me oh my god, I kill it. Okay, anyhow. All right, so somebody was raising hands. Let me see. Oh, ACH. Okay. Okay, so here we have the first part where you copy the RNA is going to copy is will is copying the DNA. Yes or no?
Yes.
That
Yes.
is the
Yes. Yes.
transcription. Transcription. That is in the nucleus. Then we have in the cytoplasm when it's coming out from the nucleus. Actually, the RNA messenger will uh, uh, create will make a sequence of amino acids. That is going to be the beginning of the primary proteins, then folding, secondary, then tertiary, and quaternary. So they can stay in the cell or they can go out of the cell, depends what the body needs. All right, so actually I want you to have two images in your mind right now. Let's travel inside the nucleus. Travel inside the nucleus. See one rope copy another rope. Huh? Okay? And that is called a transcription. Transcription is going to copy, it's going to transcribe, it's going to copy the, the recipe from the DNA. The mother and father of all the recipes are in the DNA. You decide to make or cook a protein, the RNA messenger is going to open just the recipe that you want. You don't want to open all the recipes. You want to just copy one recipe. So the DNA is going to expose, is going to give you the recipe of that piece that you want. That process of copying this recipe is called that transcription. And then when you have the recipe already in your hands, you're going out in the cytoplasm to look for the ingredients. What are the ingredients? The amino
Amino
acids.
acids.
So when you are in the cytoplasm, you're going to have your list that is made by the RNA messenger, right? and they are going to put amino acids to complete your recipe. And that is called that
Translation.
translation. Translation. Is that clear?
Yes.
Yes.
How to remember this? You need to remember number one, nucleus and then cytoplasm. Nucleus and then cytoplasma. Nucleus and then cytoplasm. Then you will tell me who is first, transcription or translation?
Transcription.
Transcription.
Transcription. How to remember that? Transcription with C, and then you have translation with L, right? So
Mm-hmm.
who is first in the alphabet?
Letter
C.
C.
And then
Okay.
it's going to be the L. Easy or not?
Mm-hmm. Yes.
Okay. So
Yes.
we have the RNA that is going to copy the RNA that is going to copy the DNA is called the RNA messenger. RNA messenger. The RNA messenger. Okay. The RNA messenger is going to be inside the nucleus, and then they're going to go still called RNA messenger outside the nucleus, but the action are going to be different. The RNA messenger inside the nucleus, I just going to copy the recipe I want to cook. Okay, and then when you have the recipe, is you read, oh, I need this, I need that. Okay, so and then you go to the cytoplasm and you're going to get your ingredients. You okay with that? Simple or no?
Yeah.
All right, so I want just to make a dynamic here, if we can, because I do dynamics in my class when it's residential, but when in online is for me much more difficult. But let's try. Let's pretend. Let's pretend what? Okay, no, let's not pretend. Okay. All right, there you are. Let's pretend. Um, who like to cook here?
Me?
Anna, okay, let's talk. Oh, you, Andrea, too. Oh, I love to cook. Gabriela, oh, Dionisio, all right, let's share recipes. Oh my God, I love to cook. Me, I'm telling you, surgeons, I was surgeon many years. Most of the surgeons, they love to cook because when you are cooking, it's look you are doing surgery. For me, it's like you're doing surgery. Everything clean, everything in place, everything. I'm very neat on that. And but anyhow, all right. So let's go to the kitchen, and you want to cook something nice, okay? Something complicated, not complicated, something nice that you want to have the recipe. So now you go to the book, right? To the recipe book. Let's make it old fashioned, okay? Because we can Google it this different, right? So now let's go to the book and open the page. Open the page, okay? This book basically is a DNA. And the DNA, and this book has 24,000 pages. In one page is the recipe that you want. So you copy you copy, take a pen and a paper and a piece of paper. See, I don't want to say. Okay. And that is going to give you the list. A, B, C, all the ingredients. All right. So you have all the ingredients written. That is the transcription.
Yeah,
You're
transcription.
transcribing, right? But
Yeah.
then you don't have all the ingredients. So where you go now? You're going to go to Safeway or Leonardo's or whatever your favorite store is. Okay? And then what you're going to go there? You're going to take a card that is representing the RNA messenger.
Mm-hmm.
Go to the store and you start to put the ingredients inside.
Mm-hmm.
And that is going to be called that translation.
Translation.
Once you have all the ingredients together, you can cook and make the protein that you want.
Amino acids.
Amino acids. Yes. So imagine the amino acids are floating here and there. Oh, the amino acid is coming. Oh, my list is this amino acid A, lysine or glycine or whatever, or the tryptophan, methionine, whatever other amino acids, and they are going to put it in your basket. And that is how you're going to create a protein. Simple or not. See?
Yes.
Okay, so that is all about the DNA, and we are going to talk more in detail in few more things. So one thing is you know the difference between the DNA when it's resting versus the chromosomes. Basically, it's different, yes, and no different, and it's not different. Why? Because the chromosomes are going to take that shape only when the cell are going to reproduce. When the cell are going to need to divide and reproduce, that is when the DNA take the form of the chromosomes. When the DNA, when the cell do not need to reproduce, is going to stay with a double helix DNA. And in certain moment, whatever, the cells are going to require some proteins because proteins are all over the body and proteins are required for our survival. Hormones, enzymes, hemoglobin, immunoglobulins, so many things are participating in the proteins. So in that rest stage of the cell, they are going to uncoil the piece of the recipe, opening the page of the book for you to copy the recipe that you need.
RNA.
And they are going to produce, for example, you need more antibodies, more immunoglobulins. Okay, perfect. The DNA is going to uncoil that piece and produce a lot of that. And the same, you need more hemoglobin, or you need more FSH, LH, or growth hormone, or TSH, any of these elements. If you need that, the cell will know and uncoil only the recipe that is required in order to produce what you need or what you want to eat. Okay?
Yeah.
Okay.
All right. So is that clear, please? So I give
Yes.
you the big picture because I don't like to read, right? But definitely we are going to go into slide by slide to make a summary of what we need to know.
Okay.
Oh, yes. You want the break? I want break all the time now because 12:15 is going to be the real thing. If I don't come back, yeah, look for me in the newspaper or something. So okay, all right. So let's have a small break. I will come back at 10:30. Okay,
Okay.
all right.
11:30.
11:30. So see, Mr. Otis is always alert. Excellent, Mr. Otis. I like that. Thank you, Mr. Otis. You were in the military.
Yes, Air Force.
Oh, I was in the army.
Oh, congratulations.
Yeah, I was. You were active in front line.
Well, no, not on the front line. I was in the Air Force. All I did was get the airplanes off the ground.
Oh, oh my God, that is wonderful. I was yeah, I was active and I was in the war for 3 months. Almost traumatized, but I
Yeah.
came, I came alive. But yeah, we will talk more about that. A lot of experience.
I'm I'm a prior service too.
Oh, you too?
Yes,
Oh,
sir.
excellent. Also, you know what is making action, what to make
Yeah.
quick decisions. No
I
secret,
work on
right?
Apache aircraft.
In the what?
I work with Apache aircraft.
Okay.
What is that? Battleground?
No, I work in the pad support.
Oh, excellent, excellent, excellent. Yeah, yeah. This is a very intense. So we will talk another time. I'm sure. Excellent, sir. Thank you. See you then. Okay, so let's continue. Cameras on, please. Okay, so let's start. All right, so now that we have an idea of what is a chromosome and when does that happen, so you can tell me one question here. Chromosomes are composed by genes, yes or no?
Yes.
Yes, perfect. Okay, so the chromosomes is actually, if you make a summation of all the 46, write down that 46 chromosomes that we have in every single cell of your body, in our body, are going to contain, actually is the same DNA but in different appearance. When you hear about chromosomes, oh, immediately you say the cell is dividing. Okay? Okay. So the DNA means the oxyribonucleic acid. The oxyribonucleic acid. Okay? The oxyribonucleic acid. That is what is called DNA. Okay? So the DNA is going to be composed by bases, the steps of the stairs, and those are actually the A, C, G, and T. A, C, G, and T. Remember A, T, and T, and C, G, very similar. Form. Now we have here the nucleotides. The nucleotides is basically what we call the base. So all this is the base. So look at this. Here, this is a very important picture, okay? So you are opening the stairs. So these are the steps where you put your foot when you climb into the stairs. Then you have the sugar here. Those are the bar. And the rails are going to be the phosphates. You follow me? Right? Okay.
Yes.
If you see here, each base, all this piece is going to be called a nucleotide. A nucleotide. That is the only word that I want you to remember, the nucleotide. So the nucleotide, you can say bases, but when you say bases, you're talking about only this. But when you take about nucleotide, you are including part of the rail and part of the bars in that segment. Okay, so for our course, our lecture, we are going to call basically the bases. These bases are related to this sugar and with these phosphates. Okay, sugar phosphate. Don't forget that sugar phosphate. Sugar phosphate. Sugars are the bars. Sugar, sugar bar. Phosphate, the rail. Okay, all right. So here we have the bases are going to be having another. This is question for the exam, please. Okay, so how they are linked one side of the step and another side of the step to match each other. So this match, they have a glue. They are going to make a glue. This glue are going to be basically an hydrogen bond, hydrogen. So this hydrogen and hydrogen is going to be basically linking the bases. Hydrogen, 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 hydrogen. So that is the hand that are, or the hook that are going to get attached one side of the base with another side of the base. Okay? So these are important. Why? Because in order, if you see here, the DNA the DNA is going to have this configuration, right? And they keep this configuration because the bases are attached to each other. So in order to open the area of the gene with a recipe that you want, they're going to go like this. They're going to open. They're going to expose that area. And that means that they need to separate the bases, half of the steps. And that is because the hydrogen molecules are going to be detached. So the hydrogen is the one, is like a zipper, is the zipper. The zipper, when you put down the zipper or you put up the zipper, is you put down the zipper, you open the zipper, right? You close the zipper, the hydrogens are coming back. And that is how selectively you're going to select one of these 24,000 genes, the recipe or the gene that you want to create a protein. You okay with that?
Mm-hmm. Yep.
Excellent. Okay, so forget about the purines and the pyrimidines. That is more actually, uh, 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 we don't need to know that. But what I want you to know is the hydrogen bondings. Where is the hydrogen bondings? By hydrogen bondings will be between A and T, or T and A. It's going to be between C and G, or G and C. So what is the bonding with? Are going to bond with hydrogens. This site is one of the double helix, the double line of the DNA that we saw here. See, this is the double line here. Can you see the double line here? This is the double line here. They're going to, this is a representation, but they are going to, we have one line and another line of the one half on the right, one half on the left. So these are going to be attached with hydrogens to the other side of the molecule. It's not showing here. But that is what we need to remember, the hydrogen bonds between A and T, or T and A, or between C and G and C. Okay? Excellent. All right, so here we have the base, the pairs of um, of the bases. We have the adenine, the A and T, and we have the C and G, or G and C, whatever you want. All right, so that is basically the DNA, the DNA. But at the difference, we have the RNA. The RNA is the ribonucleic acid. Okay? All right, so in this case, the DNA contain the A, T, C, and G, and the RNA is going to contain the the uh, the A is going to contain A. They don't have timing. They have C and G. Instead of the timing, are going to actually is going to contain what we call the uracil. That is the difference between the DNA and the RNA. That in bases, talking about base on bases, is the timing and the uracil. So the timing is present in the DNA, but the uracil is present in the RNA. So RNA do not have timing. They have only uracil instead. So now the adenine are going to attach not to a T, but to a U. That is an RNA. That it about, no more than that. Remember that DNA is a double helix structure, and the RNA is only one line. It's only one line. So here we have the ATCG DNA, and the RNA is going to have A, U, C, and G. Is that clear or no?
Yes.
Okay. So here we have our review. Here we have the base, and then are going to appear the sugar, and the phosphate that are the rails. Okay? Okay. Now, this is a DNA. You see here the T. The T is associated with A, C with G, T with A, C with G, A and T. Here we have the sugars, and here we have the phosphates. And this, what we have in dots here, those are the hydrogen molecules who are going to attach one side of the ribbon of the to the other ribbon side of the DNA. If you notice here, there is a double helix here. Oh, this is a double helix, and that is what you see here inside. They are going to open, for example, I want to open this gene from here to here. So the rest is going to be like this, but this one is going to open. And the RNA messenger is going to come to copy the recipe inside the nucleus called that transcription. And then after that, they're coming out and form that translation. Excellent. Oh, look at that. Can you see the bases? Imagine the DNA.
Yeah.
Can you see the sugars? And can you see the phosphates, the rails, right? The rails are this is the phosphate. These rails are the phosphate. And why I put this stair? Because I think I like it. That's a very nice stair. Okay. So here we have the summary. So DNA is going to be copied by the RNA messenger, and that is called that transcription, C. And then the RNA messenger is going to make the sequence of the proteins, primary to quaternary protein, that is called that translation. That translation. Translation is going to happen. When ends the translation? The translation ends when you have the primary protein. The primary protein. You okay with that?
Yes.
Yes.
Excellent. So we have 46 chromosomes, and we are talking about here about the cell division, right? So you have here 46. You and I, we have 46 chromosomes. We have 23 chromosomes from father, and 23 chromosomes from the mother. So total 46 chromosomes. So you share the chromosomes that are coming from the father and from the mother are going to be shared, and they are going to complete your body. So that means you have that's why you have characteristics of your father and your mother. That is one simple chromosome. One chromosome. There is that is trying to show you the recipes. That is the recipes, the ATCG. Okay. So that's what we have a gene. Look at this. If you cut the DNA, in pieces, you have a piece here. And these pieces start to coil, to coil, to coil, to coil, to coil, to coil, to coil. And there are, as a resultant, is going to create a chromosome. It's like you have a lace. Put a lace like this over your shoes, a lace, and try to roll it, roll it, roll it, roll it, without making tension, roll it. They're going to basically start to form like a chromosome. Do you notice that?
Yes.
Right? Okay. All right, so the nucleic acid. So in addition, they play a role in other situations. So the nucleic acids are going to be, for example, the ATP, 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 ATP. So there's another areas where we are going to have the nucleotides. One of these that is not related to the energy is the ATP. So something related with the DNA is not related to the DNA that produce actually ATPs. So what I want to tell you is this: why I'm talking about ATPs now, because ATPs has in their formula the adenosine. Adenosine. Remember the ATCG? A is from this is the DNA. A is the adenosine. Adenosine is part of the DNA. So this adenosine is going to be located too, in addition, in the ATPs. Please, question for the exam. ATP is not a copy of the DNA. ATP is not a copy of the DNA. ATP is going to have one of the components that have in common with the DNA, but nothing to do with DNA. You okay with that?
Yeah.
Okay. So what is the ATP? ATP is the battery, is the energy factory where you have in your body. So this ATP mostly are located in what we call the mitochondria. Mitochondria. That is where they are going to be formed. The mitochondria. The mitochondria. Mitochondria. We will see that in a few moments again. So ATP. Another thing that you need to remember about the ATP, that is the rechargeable ATP, please write it down, is the rechargeable battery. Rechargeable battery. When is charged the battery, we have the adenine, and we have 1, 2, and 3 phosphates. 1 and 2, 3 phosphates. So we have the adenine plus 3 phosphates. So that is the ATP. The ATP. Okay? So you're going to name, because this is a question for the exam, we have phosphate number 1, phosphate number 2, and phosphate number 3. So like you normally writing from left to right, 1, 2, and 3. Is that clear? Okay. So these ATPs are when you have 3 phosphates. That's why it's called triphosphate. Tri means 3, phosphate. So they have the adenine plus 3 phosphates. That is the ATP. That is the ATP is already charged. It's a charge with ready to be used, are charged. So they have energy that is ready to be used. You okay with that?
Uh-huh.
Okay. So now, when we are going to use the ATPs? When we are going to use ATPs? When we need energy. All right. So these bonds are they have a lot of energy in between. This is a bond number between the bond between the second and the third phosphate here. This bond is the one who is going to have the highest energy for the whole molecule. Please remember that question for the exam. So where is the highest energy in the ATP? The answer is between the second and the third phosphate. That is where mostly of the energy are going to be accumulated in the ATP. Okay? Where is the most of the energy accumulated in the ATP? Between the second and the third what?
Phosphate.
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 Excellent. All right. If you see here, we don't have the food around here. The food is not going to be participating in this step. So when the food energy is going to correlate with the ATPs. All right. So look at this, please. You have a nutrient here, and the nutrient, when you break it down, is going to release energy. Yes or no?
Yes.
So that energy, how is correlating with the ATP? Look at this. When you have an ATP, adenine, phosphate, another phosphate, and another phosphate, okay? So that is a charged battery, a charged battery. So when we are going to use the energy, we are using the energy of the ATPs, period, nothing else. We are going to use the energy of ATPs. And what is doing is they are going to break down this bond where the second and the third bond and release the energy, highest amount of energy. And that is the energy that we use for our activities. Okay?
Yes.
Now, Now the battery is going to be A, P, and P only. So that means the ADP. Why ADP? Because it's the adenosine. Instead of be triphosphate, is diphosphate because there are only two. Okay? So that is the uncharged battery. It's not charged. So all the charge are gone, is gone. So we need to recharge the battery. How we are going to recharge the battery? Recharge the battery, we want to bring one phosphate to complete the ADP. To complete the ADP. So now we can say the ATP is recharged again. So, but for to bring that phosphate, to this other phosphate to create the ATP, that need an energy. That energy is coming from the food. So the food is going to be used to bring a phosphate to recharge the uncharged batteries, the ADPs. We okay with that or no?
Yes.
Everybody's clear on that? Okay, so that's why I'm telling you the food is a source of energy, yes, but that energy is not used by your body right away. That energy that is provided by breaking the bonds of the nutrient, glucose, fat, etc., that energy is going to be used to recharge the ATPs that we have in our body, and that is to bring the phosphate to complete the triphosphate. So you have ADP at the beginning, you bring a phosphate to complete the ATP.
ATP.
Is that clear or no, please?
Yes.
Yes.
Okay. All right. So please don't. Now you are more elaborated. So you don't tell me that food is energy. Yes, but energy for what? To produce ATPs. And are the ATPs that we use as energy in our body. Okay?
Okay.
Excellent. All right. So we have here the monomers basically are going to be the bases, the nucleotides. So there is some difference here that they're going to ask you in the exam. Okay, number one, please write it down. DNA is a double strand band, double. The RNA is only one. Okay, the DNA is a double helix. You can call double helix. Double helix. That means that it's like this: double, one, and two. Double helix. The RNA is only one strand of line. Only one. Only one. This is D. Remember, D double. R is going to be one. RNA one. Okay? So both the RNA and the DNA are going to be, RNA is one strand, as you see here, open eyes, open ears, and the DNA double strand, open eyes, open ears. What we have in common is the phosphate. RNA have phosphate, the DNA have phosphate. They have nucleic bases, yes. Nucleic bases, yes. So the only difference is that they have the ATCG. The RNA is going to have the AUCG, and the DNA is going to have the ATCG. The one who is different is the timing. Timing is in the DNA, and there's no exist in the RNA the timing. Instead of that, we have the uracil. The uracil. Okay. Now the sugars. The sugars, there is another difference between the DNA and the RNA. The sugar is a ribose, but the sugar in the DNA is the deoxyribose. So that means they have like six carbon elements. That is different from the ribose that contain only five. So you don't need to know that, The only thing I want you to remember that the sugars are different from the DNA and the RNA. The sugar for the DNA start with D, DNA, deoxyribose, and the sugar for the RNA start with the R, the ribose, the ribose. The phosphates are exactly the same between them. You okay with that?
Yes.
Is that clear? You want me to repeat something or no?
No, it's clear.
It's clear. All right, so there you are. The RNA, they don't have the timing. It's written there. No timing, but instead of that, we have the uracil. You have A, G, C, which is missing the T. The T is replaced by the uracil. There is no timing in the RNA. Only R that is going to be replaced by the uracil. Okay, so we have the four type of bases. The adenine, this is the DNA, deoxyribose, right? Nucleic base or base. Don't get, confused. Nucleic base is the same to say base. Base is base, base, base. Nucleic base, whatever. Base or nuclear base are bases. The steps of the stairs. All right, so that is a summary, and that is about our DNA. I want to ask you something. Where is coming from the amino acids in the cytoplasm? Remember, you go to Safeway, to the market. So after the transcription, you're going to go to have the translation, right? Where is coming from? I'm sorry? Sorry? Somebody answer?
It's the messenger RNA.
Yeah, all right. But those amino acids that are floating in the cell in order to complete the RNA messenger translation. My question is, where are coming from those amino acids? These amino acids are coming from where? From heaven, from magic, like magic, boom, appears like.
Digestive proteins that are they're going to their monomers. Amino acids are absorbed into the intestines.
Cristina, oh, excellent. In one word, is from your diet. Got it? Excellent, Cristina. Right? From your diet. So you need to eat those proteins in order to the cell be able to produce the hormones, the immunoglobulins, the enzymes that your body needs. So we are closing a big circle here. So everything is going to be correlated or related to each other. So now you eat in your diet, you have amino acids, right? You break it down, they're going to be absorbed in the form of amino acids. These amino acids are going to be absorbed as a monomer to the bloodstream. And then they're going to get into the cells. And then the DNA decide, oh, I need these ingredients. Where is my market? Where is my Safeway? It's in the cytoplasm of the cell, ready to pick it up and produce the protein that you need. Some of these proteins are going to stay in the cell. Some of the other proteins are going to go out of the cell according to what you need. Okay? The number of who are actually, where is going to be produced those ATPs? Those ATPs basically are going to be formed inside the mitochondria. The mitochondria. Everybody knows what's mitochondria, right? Basic biology, right? Mitochondria is the the engine of the body. Inside the mitochondria are going to produce the ATPs. And then the ATPs, they can go out of the mitochondria to whatever we need to use for. Okay? Is that clear or no, please?
Clear.
Okay.
Okay. All right. All right, so now another thing that, so you can tell me, for example, this cow, goo, goo, no, cow, GG, whatever. What is this? This sequence is RNA or DNA?
RNA.
RNA.
RNA. Why you can tell me that?
U,
Because they have
uracil.
the
There
U, but this
It's got uracil.
excellent, the at cac, whatever you call that, that is DNA or RNA.
DNA.
DNA.
Why? Because they do not have the uracil. They have the binding. Beautiful. All right. So another question for the exam, please. Another question for the exam is why the DNA has two strands? Why the DNA have two bands? Why the theory is the DNA have two bands to protect each other, to cover and protect. So that is the theory that we actually have so far. So that's why we have two strands of DNA on the right, on the left, and like a spiral stairs. Okay? All right, so another old news already. Old news. The A and B are going to be linked through hydrogen bonds. The C and G are going to be attached through hydrogen bonds, right? So those are hydrogen bonds. Hydrogen bonds. So now you can tell me this. If the primary strand of this, what could be the complement of this? This is the one side of the DNA. So A should be attached to whom?
T.
T,
Keep going, keep going.
G.
G,
G.
C.
C.
G.
G. C.
A,
A. T.
T,
T.
T.
T,
A.
A.
A,
A.
A,
G.
G,
G.
C,
C. C.
T.
C. T.
A,
A.
T.
G,
A.
A.
G. A.
And this is not the same as this.
No.
No? What do you mean no?
Oh, I think it is.
It is the
What
same.
is the same?
Oh yeah. Well, sorry.
This is the answer of what you just said.
Yeah.
Okay?
Twins.
Okay, we got it?
Yes.
Yes.
All right, so look at this. Oh, what is this?
RNA.
Exactly. Very good. See? So, first of all, what happened is the DNA that is in blue is going to uncoil, 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 uncoil. There you are. And then these red bonds are going to be the RNA messenger, who is going to do what? Copy the complements. So, for example, we have C are going to be G, A and T. So they are going to create the sequence.
Transcript.
And then they go to that is the transcription, right? Transcription, transcription, and then ready for that translation. We got it?
Yes.
Beautiful. So now you know DNA and RNA, genes and chromosomes. Did that change your perspective about DNA that you had at the beginning?
Yes.
Excellent. Okay, so now talking about the chromosomes, they want us to go over this. Look at this. So imagine you are in your city. Which city you're coming from? Hayward, from San Mateo. Okay. So, all right. So town are going to be all the cells on the body. For example, San Mateo. All the houses are. San Mateo is the body. Is the body. Is the body. San Mateo. And all the cells are the houses. Okay. So the library of the city is the nucleus. The library of the city is the nucleus. Let's go inside the library. Inside the library, you're going to find 46 sections. 46 sections. These sections are going to be representing the chromosomes. Okay, it's another way to see it, right? So chromosomes. These chromosomes, each section, they have many books. So each of the sections have many books, and actually we have each big section. Big section is a chromosome. The chromosome, they have many books, and these books are the genes. And when you open the book, one of these books are going to contain the recipe that is specific for the protein. ¿Me okay with that? Okay. So to complete this portion is we have the transcription is the rewriting. Transcription. Transcription happen in the nucleus. C is first than L. So transcription, and then after that, outside in the cell, outside in the cytoplasm, are going to be the translation. Translation. Okay. All right. So that is basic. Oh, we are going to talk about mutations, but definitely I need to stop here because we are going to continue after the what? After the interview. Okay, can you wish me good luck, please?
Good luck.
Good
Good
luck.
luck.
Good
Good
luck.
luck.
Okay.
You got it.
Bye.
Yeah, for the
Yeah.
benefit of accreditation.
If you don't
That'll
see me
work.
back, just send me flowers, something. No, I'm just kidding.
You'll be all right.
Okay, gracias. All right, so I will see you at 12:45. That is basically I need to prepare material for that. Start at 12:15 with me, and then I'm going to finish at 12:45. So you will see my face, big smile, or yes, no, no, you cannot see my face. Okay, no, I just kidding. All right, so see you then. Muchas gracias.
Good luck.
Thank you. 10:45. We coming back. By the way, I'm going to close the whole presentation,
Okay.
but the recording is still running, so it's going to be running. So then I'm going to come back. Okay. All right. So see you then. If there is any question, please call me. Okay. Bye.
Bye.
Bye. 1, 2, 3. Bye. Hello, everyone.
Hello, teacher.
Hello, hello. Everything was good?
Yes.
Okay. Thank you. Thank you for your patience. Oops. Okay. All right. So, well, I'm not going to have a break. I just finished the interview, so let's continue because we have a long class and we need to do the rest. A very important topic, by the way. There is something that is going to change your perspective and that you're going to be enjoying very much about how to be a nurse and how we start these small steps I'm going to give you today. Okay, so let's continue. I'm going to open my PowerPoint. I'm going to do the presentation, entire screen, hide. Okay, let me see where is the PowerPoint here. Can you see my screen?
Yes.
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 Okay, let's continue. Okay, okay. So once last thing I want just to remark here is during the translation. If you see here, you have the transcription. This is the at the beginning the DNA is going to unfold the area of the gene that is required for that moment in that cell. They are going to come the RNA and they do the transcription. Then they go out into the cytoplasm, this RNA messenger, and they are going to have this complement. So if you see here, for example, we have GU, this is UUI is going to be the complement for the DNA. So each of these is going to be carried by a ribosome. So what I mean is this. Look at this. So you go with a recipe to Safeway, and you have a list. You have your cart there. So and there are going to be other helpers, like other carts that are going to bring you the amino acid to the sequence. So these cards that are helping to do, let's put it, the fastest shopping, these cards are coming, these cards are going to be the ribosomes. It's like you staying in place and you say, I have my list here, and everybody is helping you to bring the ingredients. So they are coming with the cards, and those cards are the ribosomes. And what is inside the card? The ingredient, that is the amino acid. So that is the role of the ribosomes. The ribosomes, what they're going to do is to are going to participate in the translation of the RNA messenger. Okay. Any questions so far, please?
No.
Okay, so let me see what. Okay, so where I am, just to be sure that it's not skipping anything. Okay, perfect. All right, so let's talk about the gene expression. The gene expression is the process where it's going to unfold one of the genes in order to express the protein. So the unfolding process of that piece of the DNA that is a gene are going to produce when they are going to unfold is they want to express some actually protein. That is called the gene expression. Okay? Okay, so now, all right, so remember, the function of the protein depends on the 3D shape. So some 3Ds are functional, but all quaternary are actually functional. So, shape, and that shape of this protein, can you see the foldings and unfolding the of the protein, tertiary, quaternary protein? That everything depends of the amino acid sequence. So, in conclusion, the shape that you require for that protein depends of the sequence of the amino acids, the type of amino acids. If you have a different, if you have a different amino acid that is not in the recipe, that is going to change the shape of the protein. If you change the shape of the protein, probably it's not going to work. It's like you are having the key of your house, and for some reason, the keyhole are actually the lock of the door is changed. So the key is not going to work anymore because the shape of the door lock are actually is different. And that's what happened with those proteins who change. When the question is when they're going to change, they're going to change because they can change, for example, the sequence. There is a protein that have a very specific sequence, and they have a very specific type of amino acids. So just remember that we have 20 different types of amino acids, and these amino acids they can have a different order, different number. They can have hundreds or they can have thousands of amino acids, and a variable is going to change to the number and the type of amino acids that are immersed on that. Now, so they have keyword here is a specific sequence, a specific number, and a specific order of sequence of amino acids. When these amino acids are going to change position for whatever some reason we are going to explain now, the protein start to be is different. It's, for example, you have a recipe for to cook something, and you need to put the onions, the garlic, the salt, the pepper, then the meat or whatever. And but instead of salt, the recipe now they said you need to put sugar. So the dish is going to be completely different, yes or no?
Yes.
Okay. So now, for example, let's talk about the insulin. Or more simple, the growth hormone. Growth hormone. Growth hormone. The hormone is a protein. Is the key. And the receptor is going to be the door lock, the door. Okay, so the growth hormone they need is the key. They need to match exactly with the receptor of the cell membrane. The receptor is another protein, so protein linked to another protein. So when they match the shape of the growth hormone, that is a protein that have a shape that is representing the key, they're going to put the key in the door and they need to match. The key in the door will be the receptor that is another protein. And if they match, you can open the door. If they match, the growth hormone start to make effect to grow the cell. Okay? All right.
Okay.
So that shape of the key and the keyhole are going to be they need to match each other. And if there is some changes of the sequence or order of the amino acids, that is not going to match anymore. So the growth hormone, for example, will not make any effect. You okay with that?
Yes.
Okay. Now, all right, so for that, we need to talk about mutations. What are mutations? Okay, mutations basically can happen so mutations is this. Okay, to make it simple. You have, for example, amino acid A, B, C, D, E, F, G, H, I. And for some situations that I'm going to explain to you in a few moments, that is going to have a mutation, this sequence is going to be changed. Instead to be ABC will be, for example, A, F, E, C, D, whatever other configuration, G, F, H, I. So if you see the order is different, it's when you're going to make a cake, you need to put, for example, you need to have the flour, the eggs, then the milk, right? And then you're going to put the sugar, right? But if you change the order, so that's why I don't like to cook sweets, because sweets is like very precise. If you change one ingredient before the other one, that is going to mess up the whole thing. So, and that's what happened with mutations. That mutation, what happened is that the sequence is going to be altered. The sequence is not going to be the original recipe, and that is going to give you a different protein, means with a different shape, and that protein probably is not functional anymore. That protein is not functional anymore. Okay, so mutations can happen in some diseases, but there's some spontaneous mutation that happen every day. Right now is happening mutations in your body. Yes, it's happening. So these mutations are very mild mutations. So why? Because the body are going to compensate those changes. So it's not basically a big or dramatic changes. But there's other mutations that can produce severe problems. For example, there is mutations of breast cancer. Breast cancer is the BRC9, that is a chromosome. So that means breast cancer, no, BCA9. So that means that the chromosome for breast cancer is in chromosome 9 that contain some genes. These mutations can happen spontaneously, can happen from genetic predisposition. They can happen for especially radiation. For example, you have radiation when you go to the sun, you have radiation. This radiation, especially in white, very fair skin person, are going to produce actually changes or mutations on the skin, and that can lead into cancers, melanomas, skin cancers. So mutations are there's spontaneous mutations, and they can be severe consequences on those changes on the proteins. All right. So points of mutation, we have changes in the single base, nucleotide base on the DNA. So instead to have A, you have C. So that is already mutation. So that is something different from the original recipe. And that can happen. And that can lead into some problems. Most of the time, these spontaneous mutations are mild. They're not going to make any effect or severe effect in the body. All right, so for example, we have some diseases that are going to, for example, we call the cystic fibrosis. Cystic fibrosis. Okay, so you don't have escape on this. Huh? You will see that in MedSearch in Pharmacology 1, Pharmacology 2, and cystic fibrosis. What is cystic fibrosis? Cystic fibrosis is a genetic disorder. It's a recessive genetic disorder. So what is happening here? This, you have mucus. You and I, we are normal. We don't have cystic fibrosis. And you have mucus. Remember the pseudo-stratified with the mucus and all that? That mucus have some thickness, right? So this mucus normally our body have enzymes who are going to break down the mucus to make it more easier to eliminate. In other words, those enzymes are going to make the mucus more thinner, easier to eliminate. In this type of patients, what we have is a mutation. This mutation are going to make that enzyme not be present or decreasing in number. And what is an enzyme? Enzyme is a protein. That enzyme that's supposed to break down the mucus to eliminate faster and easier is not going to be present. So what happened? Your mucus become very thick, very thick. You cannot eliminate anymore the mucus from the bronchi. They can lead into infections and pneumonias. Okay, so that is what is cystic fibrosis. Cystic fibrosis will be a mutation of one of the genes that produce that protein called enzyme that break down the mucus of the secretions of the respiratory tract. If you have a mutation that in that gene, the enzyme that is being produced is not going to work because of the recipe is being changed, and the enzyme that is not working is going to produce what we call cystic fibrosis. Cystic fibrosis. In conclusion, what is the cystic fibrosis? Cystic fibrosis is the thickening of all fluids in your body, not only the respiratory tract. The rest we will talk about med surg, but the main problem will be the respiratory tract mucus. You cannot break it down. Right now, you have that enzyme normally. You have that enzyme who is going to break down the mucus in more thinner, more fluid, easier to eliminate. But in cystic fibrosis, that enzyme is not present. Why is not present? Because the gene cannot produce the same enzyme because the recipe was changed. Why? Because of mutation. Why? Because the sequence of the amino acids changed. We okay with that?
Yes.
Okay. All right. Phenylketonuria is another thing that there is the lack of an enzyme that cannot be metabolize the phenylalanine, one of the amino acids. So these amino acids are going to be accumulated because they cannot be used by the body because there is an enzyme that is not present because of the mutation. And that basically can produce problems of, especially in Jewish population, elderly people, younger people. They live about 11, 12 years old because of this amino acid is needed for other we need it for other functions, other activities in the body, especially in the brain, because these people mostly they have what we call mental retardation, and they have enter into kidney failure and die. So that is because of the lack of one enzyme. So I don't want you to remember just phenylketonuria. I want you to see the concept. The concept is that if you have a mutation, the protein that is actually represent that gene, it could be an enzyme, or it could be an hormone, or it could be antibodies or some immune cells. So it could be anything, right? Anything. And that, in this case, actually can lead into death of the patient. Okay? All right, so there is basically, we have here schizophrenia and bipolar. Schizophrenia is known, schizo means division. Phrenia means personality. So schizophrenia means that you have a divide personality. So, and this schizophrenia, they have two factors, the genetic factors and environmental factors. Environmental factors. So if you live in a dysfunctional family with domestic violence, addictions and drugs and all that, there is actually a very, it's put higher risk to have some mental disorders. if you have companion with genetic predisposition, that means there is family who is running but more easily schizophrenia than other families, and that is a genetic component. A genetic component plus the bad environmental factors are working together. Okay, please, can you give me a couple minutes, please?
Yeah.
Thank you. My apologies. So let's continue. Okay. All right. So schizophrenia is it's a mental disorder that it can be affected by two things. So it could be some mutations happening in the spontaneously that make you more predisposed or higher risk to have schizophrenia. What is, I'm not trying to teach you schizophrenia or phenylketonuria or cystic fibrosis. What I'm trying to tell you is the main concept is that anything that is going to make change the sequence of amino acids can affect many things or can affect what we learned in the previous class, production of some antibodies, immune defenses, production of some enzymes. We have 1,500 different type of enzymes every single cell. And these can be, for example, enzymes we need for to produce ATPs, enzymes we need to produce proteins, enzymes we need to break down the carbohydrates, enzymes we have for everything, right? So if some of these are defect or have a problem, that function is going to be lost. Okay, so that is the point about these three pathologies. All right, so now let's talk about the ATPs. The ATPs, as you already know, is based on the A adenosine and the three
Thymine.
triphosphates, right? Excellent. Phosphate. So adenosine triphosphate. This adenosine triphosphate Adenosine is one of the ingredients that we have on the bases, right? But that doesn't mean that this DNA is ATP is not a DNA. Okay, so they share one of the ingredients. That's all, and the ATP. So we have here phosphate. We have adenine and one phosphate, and another phosphate, and you write it down 1, 2, and 3. The highest energy for this ATP will be between the phosphate 2 and 3. And the ATP, when they lose these phosphate, are going to turn into ADP. But the ADP, if you add one phosphate from the energy of the food, of the diet, that is going to turn into ATP again. So that's why we call this is a rechargeable battery. A rechargeable battery. You okay with that?
Yes.
Okay.
Yes.
Okay. Let's see. All right, so this energy of the ATP, look at this. This is a nice picture. This is 1, 2, and 3. Here is where we have the high energy bond, and that is mostly what we use. Okay? All right. So this energy can be used for many things. One of these is going to produce another molecules. For example, to produce hormones or to produce other enzymes. It can be converted to mechanical energy. These ATPs is going to be used when you move your hand, your arm, your body, you walk, you jump, you eat. All this mechanical energy is going to come from the ATPs. These ATPs can be transformed into electrical energy, where the electrical energy is happening in the nervous system. The nervous system, in order to transport, I will tell you very fast here. Look at this. This is the nerve, and we have here proteins. Proteins. Okay, proteins. These proteins, these proteins are called, when I proteins in the nerve cell are going to be called neurotransmitters. Neurotransmitters. What are those? Are proteins. Neurotransmitters. Example, for example, the acetylcholine, ACH. Example, the adrenaline, just to give you some few names. And why do we need the electrical impulse here in the nerve cell? These ATPs are the ones who produce some electrical disbalance between the out and the inside the cell. Just not to go deeper on that, the electrical impulse is going to go like this. They're going to start in the nerve cell, and they're going to be transmitted through the axon, electrical impulse, one and another electrical impulse. So this electrical impulse is like electricity passing through the axon. 1, 2, 3, 4. Each number represents a wave of electricity. When this electricity is happening here, this is going to move these neurotransmitters through the axon. So one wave of electricity is pushing forward the neurotransmitter. Another wave coming of electricity is going to be even pushing forward and more. And another electrical impulse. So the neurotransmitters are going to be pushed by this electrical activity, and then they're going to be released in order to continue to the next nerve cell. That is what we call the nervous transmission. And this electrical impulse is needed in order to transport these neurotransmitters, adrenaline, acetylcholine, we have GABA, glutamate, etc., many others. And for that, we need ATPs. So in conclusion, if you don't have ATP, your nervous system cannot work properly. And you can tell. If you are weak, you cannot think, you cannot concentrate. If you are hypoglycemic, you cannot focus. Or no? Is that correct?
Yes.
Excellent. Okay, so let's keep moving here. I'm not going to go on that. All right, so here we have the ATP is a rechargeable battery. That is what is important. Rechargeable battery. So when is charged the battery? The battery is charged when they have the three phosphates, so ATP with T as in Tom. And when this uncharged is when they lose one of the phosphates. So now it's going to be called ADP. ADP. In order to recharge the battery, you need to put or to bring a phosphate to the ADP. So you need to bring a phosphate to one ADP because they have two only. But to bring that is need to have some energy. That energy is coming from your diet, from the food. So in order to put the phosphate and recharge the battery, you need to have your food, your diet, energy coming from your diet. Is okay that? We okay with that? It's clear?
Yes.
Okay.
Yes.
All right. So mostly of the ATP, sorry, basically all ATPs are going to be formed inside the mitochondria. Inside the mitochondria. Now, there's cells in the body who have different number of mitochondria. Some cells they have a dozen of mitochondria. Another cells they have hundreds of mitochondria. Some cells they have thousands number of mitochondria. And that depends how much the cell needed. For example, we have the liver. The liver. The liver. The liver is the big factory of your body. Yes, it's the big factory. The liver have 512 functions. 512, 5112 functions. I will make you memorize 4 or 5 of them. But at this moment, you know if a factory are going to have so many activity, so many functions, tell me that factory they need more or less energy?
More.
More energy, right? If the cells of the liver they need more energy, they will have less or more mitochondria.
More mitochondria.
More mitochondria. So different parts of the body depends of the activity and the function of activity function of the organ. They are going to need more or less number of mitochondria. Just to give you an example, what is the cell who actually has the highest number of mitochondria? And that is the ovum. The ovum coming out from the ovary. The ovum is the one because it's going to have huge transformation, huge activities, a lot of enzyme, a lot of activity, and that is even more mitochondria than the liver cells, the ovum. Okay. All right, so eat or not to eat. What they said here. So let's see. That is why we need to eat. When we eat food, molecules, carbohydrate, protein, fat, we break down those molecules to extract the chemical energy trapped in them. When the ATP is used for energy to fuel functions, it gets recharged using the energy of the food. We okay that? Now we understand that very clear?
Yeah.
Yes.
Okay? Okay. So that is about that. This is a optional topic. I'm not going to go in the optional, but there is very important topic here that is the diseases. This is the second part. Okay, are you ready?
Yes.
All right, excellent. All right, so now we are going to talk about different situations. Let me see, how long is this? Yeah, okay. All right, so what is a disease? So please, I want just you to remember a few things here. A disease is an abnormal condition of the body where the functions of the system or organs are not the same. So they are going to have changes on the function of the organ. So abnormal changes on the functions of the organ, that is what is called a disease. Okay, a disease at the cellular level is that the cells are not working or are working abnormally. So they are not accomplishing the function that is being actually made of. So that unbalance of the physiology and the function of the system or organ that can lead into disease. For example, if you don't produce enough T3 and T4, those are the thyroid hormones, you will have hypothyroidism. You start to fold your hair, your nails are brittle, loss of memory, alterations on the menstrual cycle, etc. if if you, for example, you have the insulin, what it's doing is to bring glucose inside the cell, right? That is the function of the insulin. If the insulin is abnormal, or the receptor for the insulin is abnormal, the function is being impaired, and that is going to produce a disease called diabetes mellitus. Got it? Okay. All right. So now that from now on, that is what I'm going to tell you is things that are going to really change your perception about health and disease. All right. All right. So we have here hereditary disorders, nutritional disorders, infectious diseases, metabolic, immune disorders, degenerative cancer, and psychiatric disorders. Okay. So you as a nurse, so let's start this. You need to, first of all, when we are going to see a patient, we always look what is the most common. What is the most common? Before you go into more complex and detailed and rare situations, you need to always look for what is the most common. Okay, so diseases can be organized in or classified in different ways. We have hereditary or genetic disorders. Hereditary or genetic disorders will be, for example, the cystic fibrosis that we mentioned. It could be, for example, Down syndrome, Down syndrome, Down syndrome, and other syndromes, Leclerc-Duchat, Patot, whatever, DiGeorge, whatever. So some genetic disorders. These genetic disorders mostly happen in a child, in a newborn and child, newborn. Okay? Now, another problem that you have here, and we are going to nutritional in a few moments, is the infectious diseases. Infectious diseases, which group of the population are most commonly having infectious diseases? Yes or no?
Child. Oh, yeah.
Right?
Senior.
Children, they have a lot of more common, but that doesn't mean that adults we don't have infections. We have infections, right? Elderly people who have infections, they have infections too. But the most common, you will see simple cold, flu, viral infections, whatever. Kids are the most common. So what I'm trying to tell you is this: if you see a kid coming into your office or coming into the ER, the first thing you're going to think about is probably we need to check for infections, because this is the most common presentation of disease in this group of population. Metabolic disorder. Metabolic disorder, we have the how you process your cholesterol, for example, how you process your calcium in your body, how you process your diet. So metabolic disorders are going to be, for example, gallbladder stone. We have kidney stone, kidney stone. We have, for example, thyroid problems like hyper and hypothyroidism. And tell me, do you know somebody with gallbladder stones?
Yes.
Tell me, this patient is middle age, elderly, or young?
Mostly middle age.
Middle age.
Middle age.
Do you know somebody who has kidney problems, kidney
Yes.
stones?
Yes.
Yes.
Yeah, is middle age, young, or very old?
Middle age.
Middle age. Person who have thyroid problems, you see that is basically very young or mostly very early or middle age?
Middle age.
Middle age. So when you see somebody with these problems that are called metabolic disorder because it's going to alter the metabolism of the body. Gallbladder stones are going to be the presence of cholesterol in your gallbladder. Okay? So can you give me a favor, please? A favor is, can you put your hand under your table? Can you? Do you? Yes?
Yes. Yes.
Everybody, please. Do you feel
Yes.
that gum? Can you feel that gum under the table? Do you see it? Right? Do you touch it? Who found the gum?
Not.
All right. All right. So that gum is exactly the same texture that a cholesterol. Yes. Why I know that? Because I open cadavers. I make dissection and I do necropsies, whatever I did, right? So, and I open and I found cholesterol deposits, and those are exactly the consistency and the texture of those. So these gallstones on the gallbladder are made of basically from cholesterol. So that is because you're eating too much fat, too much cholesterol, and cholesterol is eliminated through the bile. If you have too much sugar in your tea, put a lot of sugar. The sugar, there is a moment that will not dissolve. And the same thing, when there's cholesterol too much in the bile, the bile cannot dissolve the cholesterol anymore, and the cholesterol precipitate as gallstones. The same for the kidney stones. If you have patients who do not eliminate as much calcium than others, they can have a risk to produce calcium stones. So, very common mistake that they think that the gallbladder are calcium stones. No, it's not. The calcium stones are in the kidney, and the cholesterol stones, now what you call, because when somebody say cholesterol, they say fat. Fat is like a loose fat. No, it's not like a loose fat. It's very dense fat, as the gum that you found under your table. Okay? All right, so we got it?
Yes.
Okay. And the other one I want just to go over is the degenerative diseases. Degenerative disease. Degeneration means to destroy, to not be as the same as before. And that degenerative disorder will be, for example, the
Alzheimer.
atherosclerosis. Excellent, very good. Atherosclerosis. What is atherosclerosis? Athero in medical terminology means deposit of,
Deposit.
of plaque, or plaque. Plaque or deposit of what? Of cholesterol.
Cholesterol.
That is going to basically invade the walls of the vessel and destroy the vessels. Degenerative disorders are going to be, for example, Alzheimer, that can include psychiatric disorder,
Yeah.
Alzheimer. So those are degenerative disorders. So you can tell just looking at the age of the patient. And by the way, the another degenerative will be considered cancers. Cancers. Cancer. Tell me, cancers are most common in very young, middle, or elderly people?
Elderly.
Elderly.
Elderly people. So now you have three main, pathways. One, what is the most common problems in kids? Basically, infections.
Infections.
What is the most common problem in adults? Metabolic
Metabolic.
disorders.
Or
What is the most common problems in elderly people? That
degenerate.
degenerative disorders.
Disease.
So that is going to give you a guide. So basically, you need to follow the whole nursing process in order to actually investigate this. But the first impression is to start guiding yourself on what you need to focus on. If the patient is a kid, you're not going to look for hypertension, or you're not going to look for cancers. Kids can have cancers too, but it's not that common. Most common than that are going to be infections. And besides that, what kind of infections the kids are going to have. Number 1 and 2 are going to be urinary infections in girls, especially little girls, and respiratory infections, upper and lower respiratory infections. And based on that, you need to start alert without actually overlook other components, but you in your mind you already know that the most likely situation is this. You okay with that?
Yes.
Yes, sir.
All right, so that is about that. Just remember, homeostasis is the uh, uh, keep how a body keep the internal balance, and how to keep the internal balance is talking about the nervous system and the endocrine system. Okay, nervous system and endocrine system. You okay with that?
Yes.
Okay. So hereditary or genetic. So for that, I have time to do this. Hereditary, for example, we was talking about schizophrenia. Okay, let's make an example of this. I want to see faces. Okay, listen to this. All right. So do you know somebody who has schizophrenia?
Yes.
Neighbors or friends or somebody, right? Okay. It's very degenerative disorder, right? So a lot of radical things happen. All right, so let's suppose this. Look at this. Why you are not schizophrenic? Why other people have schizophrenia and not you? The colloquial way to say, oh, because you are stronger, right? So people say that, oh, it's stronger. They don't have schizophrenia. So, but what? Let's translate into medical terms. So, what does it mean? All right, I will give you an example. You have uh, a family, 2 kids. They are twins, identical twins, identical twins, identical twins. They share the same genetics and characteristics are identical. You okay with that? Okay. But the family who has them, the family are having running some schizophrenia in the family. Okay, both kids are going to suffer schizophrenia. The answer most is not necessary. So why? How we explain that? Look at this. If you have, for example, your threshold. Let's make a this threshold. This threshold. Threshold. This is how much you can stand in order, for example, problems in the family, violence, whatever. So you have this threshold. You can stand more those problems. But the person who has some genetic disorder or family disorder with schizophrenia, the threshold is going to be this, very narrow, much more narrow. So there's some predisposition. The patient is not schizophrenic, but is more predisposed to have schizophrenia. Now, these kids, the twins, identical twins, they have the same threshold, very narrow threshold, because they have family history or threshold. They are not schizophrenic yet. Now, what happened? With this narrow threshold, these 2 kids were adopted. One goes to a family who is very functional family, very kind, very lovely family, etc. And the other kid go to a family who is very dysfunctional family. All right. So 2 opposite situations. So A is the first who go to a family who is normal, let's put it that way, and B is the other twin that is going to be in dysfunctional family. Which one do you think is most likely going to produce schizophrenia? A or B?
B.
B.
B. There you are. So the other one have the same risk. The A have the same risk, but the environmental factors are not contributing to get into schizophrenia. So that is telling you that always and never forget that genetic disorders go hand by hand with environmental factors. You okay with that?
Okay.
Okay. Another person, for example, smokers. There's people who smoke, and there was an actor that was actually, I don't remember his name. He was saying that he was always known by smoking all the time. And one journalist asked him, "Why you smoke? Why you stop? Why you smoking so much? The doctor didn't tell you that the smoking is bad." And the actor was answering, "My doctor died from lung cancer."
Predisposition.
Exactly, that is the predisposition. Again, we got it?
Yep.
Okay, very good. Sio, you okay? You got it? Monsieur? I see you. Miss Jojo. Are you okay? Jasmine, are you okay?
Yes, doctor. Thank you.
Excellent. Aurora? Miss Aurora, okay. Oh, Super Otis, how are you doing? You okay?
I'm doing fine.
All right. Oh, excellent. Dionisio, you okay? We have two guys
Doing
only
great,
here.
sir.
Oh, three guys. Oh, there is Chia. We have three. Excellent. Chia, you was in the war too? No? No was in the war.
Just Army.
Okay. Where you was when we need you? No, I'm just kidding you. I'm kidding you. Okay. So let's continue. All right. So that is the environmental and there's many diseases that this is not I would say med search. So that's why I want to the concept. Those are just examples. All right, so about nutrition, this is kind of yes, we need to talk about that. Okay, all right, so okay, the time is getting short. All right, so we have nutritional disorders. Nutritional disorders can happen at any age. See, they can happen at any age. So in this case, nutritional disorders can be mostly affecting in what stage of our life. So if you are malnourished when you're adult, it's not the same impact that we have when you are malnourished when you were a kid, right? And
Yes.
for that, thank you, we need to remember two things here. Marasmus and kwashiorkor. Marasmus and kwashiorkor. Marasmus, please, I want you to remember this. Can you see here this kid, even with some edema, they can produce edema, swollen. Why? Because no proteins, no oncotic pressure. The water escape to the tissue. They can be swollen. So, what is the marasmus and kwashiorkor? You must remember this name, please. Marasmus is happening in less than 2 years old, and kwashiorkor happen in more than 2 years old. What is marasmus and kwashiorkor? Is protein deficiency. Protein deficiency. So please don't forget that. Protein deficiency. Marasmus versus kwashiorkor. So these actually produce a delay on the growth and development of the baby. So the baby will never reach intellectual level as should be. So never going to be the 100%. So that's why these two names are very important. So marasmus are going to affect the baby in more depth because it's a younger. So that is where mostly all more anabolic reactions occur, and central nervous system are going to be affected by sure 100%. Okay, marasmus and kwashiorkor. So you need to remember those names. How to remember that? Marasmus less than 2 years and kwashiorkor more than 2 years. How to remember that? You need to remember the words. Marasmus. Who is longer word, marasmus or kwashiorkor?
Kwashiorkor.
Kwashiorkor have more letters, right? That means
Yeah.
more
Yes.
more than 2 years. Just to remember. And marasmus have less number of letters, less than 2 years old. Okay, we got it?
Yes.
Okay. Now, don't actually get confused with the rickets disease. Rickets disease is a very important disease that is basically happened by the deficiency of vitamin D. Rickets disease. Rickets is in children, and osteomalacia is in adult. Is the deficiency of vitamin D. What is doing the vitamin D? The vitamin D have two sources. One is your diet, vitamin D, and the other one will be your skin. Your skin produce vitamin D. When you expose your body to the sunlight, you're going to produce vitamin D. Vitamin D. And what is doing the vitamin D? Vitamin D are going to absorb calcium. If you don't have vitamin D, you cannot absorb calcium. So as a consequence of that, calcium produce weak bones, brittle bones. The disease, when that happens in children, is called the rickets disease. And in adults, it's called the osteomalacia. Osteomalacia. Okay, osteomalacia. You need to remember rickets and osteomalacia. Rickets and osteomalacia. Rickets, short number of letters, few number of letters. So children. Osteomalacia, more number of letters, more letters, that is in adult. All right. So if you see here, the one of the characteristic in children, Rickets disease in this case, is the bowing of the limbs. Bowing is going to be like this. Bowing. It's not supposed to be straight, but no, it's bowing. So why is bowing? Why is bowing? You know that if you have deficiency of vitamin D, your bones are weak, but you are still growing somehow. So the weight of the head, the weight of the thorax, the weight of the abdomen, the weight of the upper extremities, the weight of the other upper extremities, the weight of the pelvis, everything is going to push against the femur. And that's why the bone start to have this bowing appearance. You okay with that?
Yes.
Excellent. All right, so here we have immune disorders. These immune disorders, we have, for example, lupus. Lupus is the systemic lupus erythematosus. You don't need to remember that, but it's SLE. Do you know somebody or you heard somebody having lupus?
Yes.
Yes.
Okay. Now, unfortunately, I will tell you. Tell me, this lupus is a male or female? What is your patient you saw?
Female.
Female.
Email.
Female.
There you are. So autoimmune disorders are affecting more female than male. More female than male. In the range of 4-1. 4-1. For every 4 women who has lupus, 1 will be male. And not only for lupus, it could be for rheumatoid arthritis. Do you know somebody with rheumatoid arthritis?
Yes.
Yeah.
And that is female or male?
Female.
Female? Somebody
More-
so male?
No, female.
Female. Okay?
Female.
And that is again an autoimmune disorder. What is an auto? Auto is yourself. Autoimmune disorder means that your immune cells are going to destroy your own body. And that is happening. Why is happening? Nobody knows yet. So these diseases cannot be cured, can be controlled. That is different. So you need to live for a long period of time. Okay? Uh,
Yes.
And lupus, why is called lupus? Lupus because they have this rash on the nose and the cheek, because the lupus what produce is photosensitivity. It's not because some magic or some spell on the person. No, no, no at all. So the sunlight is going to hit when you go to the beach where you put the sunscreen in your nose, your cheeks, and the forehead. Yes or no?
Yes.
Yes.
Why you doing that? Tell me why you doing that? Because everybody is doing that?
Sensitive.
Because it looks good? Because it's fashionable? Or what?
So you don't get
The
sunburned.
hair is more sensitive.
It's the first that got sunburned.
Exactly. That is
More
the
sensitive area.
this
The first area that got sunburned.
exactly, because the angles of your face, that is what is going to be more exposed from the very beginning to the sunlight. So that's why you need to protect those areas. So patients with lupus, when they go to the sun, they're going to have this redness on the face, nose, cheeks, forehead, and that can produce even ulcers. And that somebody in the past said, "Oh, look like a wolf." Look like I don't know how they do it, but when somebody said, "Look like a wolf," and from there they put this disease wolf, that in Latin means lupus. You got it?
Okay.
Okay. All right. So tumors. All right. So I'm going to do into tumors very. What time is it, please? Because we
148.
148.
1:40. All right. So let's talk about what is uh, really, really interesting, and we need to know very clearly this: the need of terminology, disease terminology. We have these are questions for the exam. Okay, all of these are questions for the exam, and we finish with that. And we are going to try to do some math after hour. All right. So we have disease term etiology. So this is memorization. Etiology means a study of the cause of the disease. You got that? So you must know that.
Yes.
What is the etiology of this? COVID-19. What is the etiology of this? Tuberculosis. The bacteria, Mycobacterium tuberculosum. What is the cause of this urinary tract infection? E. coli. Etiology is the study of the cause. Who is the cause of the? What is the cause of the disease? All right. Now, incidence is something that you're not going to study statistics, but that is the minimum that you need to know, because you need to read articles, and the articles are based in investigations. And articles, if you want to get updated more and more, you need to read some magazines, and they are going to talk about incidence, and they are going to talk about prevalence. So incidents are going to be the new cases in a year. And prevalence are the total cases, old plus new cases. Okay? For example, we have 32 million people with diabetes mellitus in the United States. And we have 1,200,000 cases every year. All right. the 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 new cases in a year, 1,200,000, that is called incidence. Incidence. And the total number of old, the previous past, and the new ones in that year are going to be
Prevalence.
called prevalence. Prevalence. Okay, we can with that?
Yes.
Past, prevalence, past and right now. Incidence is what happened in the last year. Okay, so that is going to, for example, what is the morbidity? Morbidity. Morbidity is actually a disease called disease. Get sick. Somebody to get sick. Get sick. What is the morbidity of diabetes mellitus? 1,200,000 patients. So this is the morbidity in that disease, morbidity. What is the prevalence morbidity? Prevalence morbidity will be 32 million. And those are actual numbers, huh? Those are actual numbers. Okay. Now, all right, so now let's go into what I wanted to do from the very beginning. Let's talk about acute and chronic. So this is a very important thing where you are going to classify if the disease is acute or chronic. And you can read that there now. The cutoff to say is acute or chronic is 3 months. 3 months. So 90 days. 90 days. Tell me, do you know somebody who has back pain?
Yes.
Yes.
How long is that back pain? More than 3 months or less than 3 months?
More than 3 months.
That is what?
Chronic.
More than chronic.
Chronic.
Excellent. You have pneumonia. This pneumonia is going, you have pneumonia today and is going to last for 10 days. That is an acute process or chronic process?
Acute.
Acute.
Acute process. You have hypertension. Hypertension is going to be acute or chronic?
Chronic.
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 Chronic. Yeah.
Chronic. Excellent, right? So because hypertension, you cannot cure hypertension. You don't cure hypertension. You are going to control the hypertension. That is different. Okay?
Yes.
Yes.
All right. Excellent. So you already know what is a, but that is going to work for about 90% of the diseases. 90%. For example, hepatitis B. Do you have somebody with hepatitis B in the past? That
Yeah.
process is going to last for about 6 months. 6 months. That is acute. But there is a specific. The majority of the diseases are going to be follow the 3-month rule. You okay with that?
Okay.
There is some
So I have
exception.
a question. So if they have hepatitis B, which leads to liver cancer, wouldn't that be considered chronic?
Yes. In hepatitis B, from 100 of the patients who have hepatitis B, 90% are going to cure after 6 months. But 10% of those patients become chronic. So I'm answering you that because I don't want to show or to make students think that hepatitis is going to be always chronic or is going to be acute. It can be acute or it can be chronic. If that is going to resolve in 6 months, acute. If there is going to be continuing after 6 months, that is going to be chronic. So that means that in hepatitis B specifically, you will have like acute is going to be 1, 2, 3, 4, 5, 6 months. But if the after 6 months continue, that means that is going to be chronic.
I can
Okay?
apply this for kidney disease as well.
Kidney, what kind of kidney disease? There's
Like unfunctional.
all right. So we have different genetic disorders of the kidneys. ARPKD, ADPKD, MCKD, multiple, etc. So multilocular. So those are genetic. When they are coming genetic, those are chronic. But you have a problem of the kidney, for example, you have kidney stones. That is more than 3 months, right? That is a chronic problem. When you have urinary tract infection, the urinary tract infection in the majority can be cured and treated and go away in about 2-3 months, 2-3 weeks, sorry. And that means that is acute. But other problems of infection of the urinary bladder called chronic cystitis, because that patient are having more than 3 months trying to fight against infection but cannot be controlled. Okay?
Okay.
All right. Idiopathic. 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 Idiopathic means, listen to this. Idiopathic. I don't know. I don't know. So in medicine, when somebody, there is like a way to say, elegant way that they say, I don't know, or that is idiopathic origin. The etiology is idiopathic. Okay, I don't know. So idiopathic related to know what is the cause of the disease. If you don't know what is the cause, that is idiopathic. We cannot tell what is the origin. That is idiopathic. Okay?
Yes.
Okay. Then we have the iatrogenic. Another question for the exam is coming. Iatrogenic means that any side effect or adverse effect caused by a medication. A iatrogenic, for example, you have nauseous because you took the medication. That is the iatrogenic side effect, or just iatrogenic. That's it. Iatrogenic. Iatrogenic could be not only for a medication, but some advice from the doctor. Some advice. If they tell you, for example, you're hypertensive and they say eat normal, that is iatrogenic. Why? Because you suppose in your diet you need to decrease the level of salt. Otherwise, you have hypertension problems. Side effects, other drugs that can produce nausea, vomiting, diarrhea, rash, etc. Those are effects or side effects coming from the treatment, the iatrogenic. Communicable disease. Communicable disease. Communicable disease, please. Communicable disease, communicable disease, communicable disease is not like gossiping. Okay. It's not like, oh, somebody has this or that. That is not communicable disease. Communicable disease is how it's going to be transmitted from person to person. Communicable disease. So they can go through the saliva, through the spills of saliva, through blood contact or intercourse, or another they're going to be for example, the little kids, they don't know how to wash your hands. I don't care what you say, bacteria. What is bacteria? I don't care, right? So I eat my apple and at the same time I'm scratching my butt, right? Or picking my nose, right? I'm putting my picking my ears, right? And then, oh yeah, my bubble, come eat. So it's like a party of bacteria and viruses, right? So this communicable disease, for example, will be a communication of fecal oral, fecal poo, oral mouth. When is that happen? When you don't wash your hands. When you don't wash your hands, right? So communicable disease, a pathway how they are going to be transmitted from person to person. Okay?
Yes.
Okay. Epidemic. Epidemic means we have another term that I'm going to add here. It's called endemia. And that is the only one I'm going to add in addition. Endemia. Endemia is the normal number of cases that happen every year. We have endemia here in San Mateo. Yes, we have endemia. What endemia is that? For example, the number of persons who have cold, a cold, a cold, right? A viral infection, cold. I don't know what is the number. It's not registered, but endemia. That is what we expect every year. In San Mateo County, we have about 700, close to 800,000 people. And let's suppose that 1% of these people are going to have, that is 80,000. 8,000. 8,000 are going, so how much? 800,000, right? 800,000, 8,000, yes. 8,000. 8,000 people every day, every year of a simple cold. Let's suppose. But if these numbers go higher, much higher than that, like we expect between 8,000 and 9,000 a year, but now we have like 12,000, 15,000, that is already an epidemic. epidemic. So it's overpassing the number of cases that you were expecting from the previous year. Pandemic is all spread around. Pandemic is a pandemic. The best example is
COVID.
COVID.
COVID-19.
Yeah.
There you are. COVID-19. Pandemic. Pan. So do you heard the word Pan American? Pan America,
Yes.
right? Pan America, pan. Pan means America, all the countries of America.
American.
Not only in the United States, all the because America is not only United States. America is all South America and North America. So those are Americas. Okay? So pandemic means that is spread out in a large area of the disease. Nosocomial infections, please, this is important. Open eyes, open ears. Nosocomial infections, nosocomial means infections that you get in the hospital. In the hospital. The two most common infections in the hospital are going to be respiratory infections and urinary infections.
Yeah.
Okay, the most common respiratory infections. The most common will be urinary when you have using catheters or something like that. Okay, but nosocomial means infections that happen inside the what?
Hospital.
The hospital. Okay, so what time is it, please?
2:03.
Can you give me 5 more minutes, please?
No problem.
Okay. All right. So this is what I was waiting all day, talking about signs and symptoms. Now we are going to learn. This is crucial for your education. All right. So we have a sign. I'm going to start with a symptom first. Symptom. What is a symptom? Somebody can tell me what is a symptom? Symptom means subjective. Not measurable. Sign is objective and is measurable. Okay?
Mm-hmm.
Now, example of a symptom. Pain. Example of a symptom is pain. Example of a sign will be fever.
Fever.
What else? Blood pressure, respiratory rate, heart rate. You can measure. That is a sign. So that's why it's called the vital signs. Oh no.
Yes.
Okay. So the vital signs it's a sign that is measuring somehow the temperature with a thermometer, the blood pressure with a blood pressure machine, the respiratory rate, just counting the respiratory rate per minute, the heart rate, counting the heart rate per minute. Fever, temperature, normal go from 97 to 100 Fahrenheit. So respiratory rate go from 12 to 20 respirations per minute. Heart rate go from where? From 60 to 100
To 100.
beats per minute. Heart rate and the blood pressure. Blood pressure. Blood pressure, the minimal normal is 90/50. And the maximum normal is 120/80. You have everything right now, huh? So this is very important to know. We okay with that?
Okay, so this parameters is more for adult, right?
Yes.
Okay.
Yes. Yeah, and are NCLEX, by the way.
Okay.
So some people say 16 to 18 or 16 to 25. No. Respiratory is 12 to 20. Period. Nothing else. Okay? All right? So now tell me one thing. Nauseous. Nausea is a sign or is a symptom?
Symptoms.
Symptom.
Symptom. And vomit
Symptoms.
Sign.
is a sign. You can measure the vomit,
Measuring.
Oh.
right? You don't take the temperature, of course, right? But but you can measure the volume of, for example, bleeding. Bleeding
Sign.
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 is a sign. You can measure the bleeding. Nauseous cannot, right? Diarrhea,
Sign.
Sign.
cramps,
Symptom. Symptom.
Symptoms.
symptom. Beautiful. Excellent. Excellent. The most common example of symptom is going to be pain. Pain. You okay with that?
Yes.
Now, now you're going to enjoy this. So, what is a syndrome? A syndrome is the group of signs plus symptoms. That is a syndrome. Okay? Now, a syndrome, for example, I'm going to give you the febrile syndrome. Febrile syndrome. Febrile syndrome. Febrile means fever. But wait a minute, you said fever is a sign. But how come is fever a syndrome? So,
Because
febrile syndrome means that are the group of signs and symptoms. Tell me, when you have fever, your temperature go up, yes or no?
Yes.
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 Tell me, do you feel weak or you feel strong with fever?
Weak.
Weak.
Weak. Do you feel headache? You can feel headache,
Yes.
right? Pressure in
Yes.
your
Yes.
eyes, and that headache and pressure in your eyes is a sign or is a symptom?
Symptom.
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 Symptoms.
There you are. So the febrile syndrome, fever accompanied by a febrile syndrome. A febrile syndrome will be the elevation of temperature that is a sign, plus all the headaches and pain and uncomfortable feeling that you have, like headache or retro pain behind the eye, right? So
Yes.
that is febrile syndrome. We okay with that?
Yeah.
Another syndrome is the pain syndrome. Pain syndrome. What is a pain syndrome? What is pain? Pain is a symptom, correct?
Yes.
But tell me one thing. If you are pain, are you nervous?
Yes.
Yeah.
Yes.
And what happened with your heart rate?
It's beating
Goes
fast.
up.
It's beating fast.
Mm-hmm.
So you can recognize a patient who is telling you have pain, the heart rate go up, the blood pressure go up if you have pain, your respiratory rate go up if you have pain, plus the pain itself is a symptom. So all this group of signs and symptoms is called the pain syndrome. Nice or no?
Yeah.
Yes.
Yeah.
Okay. And the last thing, what is a disease? A disease is a group of syndromes. You got it?
Yep.
Yes.
Okay. So I hope that this changed your perspective about how we talk in medicine. Basically, I've given you the pathway how we can talk about these problems. And the last that is a review basically. Oh, okay. So two lessons, please. Yes, we have primary disease prevention, secondary prevention, and tertiary prevention. The primary prevention is going to be to decrease the risk factors of, for example, lifestyle. For example, primary disease prevention, prevent the appearance of the disease. Going to do your exercises, do your good diet, do your vaccinations, not smoking. That is primary disease prevention. The secondary prevention, you can write it down there, is when you go to the annual visit to the doctor once a year. So you have that visit to the doctor to detect earlier. Detect earlier. To detect. Detect. No. Detect. Detect earlier a disease. This is to prevent the disease. And that is to, tertiary is to treat the disease. Okay? Okay? Yes?
Doctor.
Yes.
All right. And the last thing here. Mode of transmission of defects. This is the last slide. We have direct contact, touching, shaking hands, kissing, sex with an infected person. Indirect contact, airborne. Somebody is sneezing. So when you sneeze, how you sneeze? You're going to sneeze like this, right? Yes or
Yes.
no?
Yes.
Why you sneeze like that? Because everybody doing it?
No, so your hands don't spread them.
Exactly. Don't do this. You sneeze and you wash your hand, your face. Don't do that. That is very, very gross. Don't do that. I saw people doing that. Okay? So airborne, we have a droplet, etc. Portal of entry, and that is the last. Portal of entry is how the microbe can get into your body. Through the skin. You have some opening of the skin, bacteria can get in. The mosquito can bite you into the skin. They can produce infections. Respiratory tract, air coming into the respiratory tract. Some bacteria came in. Digestive tract, you eat something, you put the bacteria inside your body. Urinary tract, the urinary tract, especially little girls. Little girls, they have urinary tract infection because they don't know how to clean themselves yet. So, the anus is more posterior and the urethra is more anterior. If you clean from posterior, anterior, you're going to carry fecal material to the small urethra of the little girl. And these bacteria start to climb into the urethra, produce infections. In male, it's not that common because the urethra is longer. So that's why. Now, the urinary tract. And reproductive system is by sexual intercourse or sexual contact. Okay. We okay? We finished. Sorry for that. I take 15 minutes more. But I think, do we learn something today?
Yeah.
Yes.
Yes.
We change our perspective about things a little bit more, right?
Yeah.
Yes.
All right. Anything that you want to clarify or anything you want me to go over again or to make it more or explain in different way? Everything is clear?
Everything is clear.
Okay. So what I teaching you is exactly what I want you to remember. You're going to have results very soon. I promise. Second, when is going to be our next meeting?
Wednesday.
Wednesday
Wednesday.
or Tuesday.
What Wednesday? What
Tuesday we
Wednesday?
have
What Wednesday?
Tuesday is tutoring.
Tuesday.
Oh,
Okay.
Tuesday.
Tuesday, 9 p.m.
Yes, 9 p.m. All right. All right. Muchas gracias.
Dr. G, sorry,
Go ahead.
just
I'm
one
going to
question.
stay until the end. If somebody want to ask questions or something, I will stay until the end. All right.
Okay,
So
thank you,
meantime,
Doc.
yes. Any question?
Dr. G, yes, Dr.
Yes, Anna,
G.
please.
Yeah, in regards with our CPR and first aid, um, this is also some of my classmates' questions, so I'm just bringing it up.
Yeah.
Does it need to be Red Cross accredited or
Wait, wait, wait,
any
wait, wait, wait.
department?
Oh, yeah. They need to be accredited by what is the company? Which is the Red Cross?
American Red
Yeah.
Cross.
Yes,
Yeah.
Red Cross.
Yeah. Yeah. I think the school is providing that, but I don't know when.
Oh,
So you need to ask when is the schedule for this event? Otherwise, you need to go. You can find online, I guess, where they can go, but they need to go the Red Cross.
All right.
Affiliated to the Red Cross. Okay.
Yeah.
Thank you, Miss Anna.
No other
Any
questions?
I have a question. If I have it, but it expires next year in March, so that's fine.
For this moment, yes, but at the time it expires, you will need to have another one. So probably it's better to have it now for the next 2 years.